This is the particular method wherein there can be the silencing of a gene. Now, you people understand what is silencing, or if I want to silence any genes, what would that mean? Will I put a mask on the gene and be like, shut up? What will be the silencing in terms of genes? Like, what do you people think? It doesn't have to be the right answer. What do you people think?
To stop the gene from doing its work, like
To stop the gene from?
doing its thing, whatever it does.
What does the gene do? You are right, actually. You are right, actually. But do you people remember central dogma of molecular biology?
Yes, ma'am.
What happens in that? Tell me.
The, information travels from master DNA to replicated DNA.
That is replication. Then what happens?
Reverse central, central dog.
You are not audible, dear.
I don't remember.
Anyone else? That is from your syllabus. No one remembers. Okay. I'll only tell you I remember. See, central dogma of molecular biology tells us how there is a unidirectional flow of information. From DNA, new DNA is made. Then from the new DNA, or you can say from the DNA, mRNA is made, that is transcription. From the mRNA, proteins are made, that is translation. Now, once the proteins are made, protein is what we require. Like, for example, any kind of hormone or any kind of gene in our body, whenever it gets expressed, it means that proteins are made. Okay, meaning translation, transcription, these things have been done. When we do gene subtraction, then this doesn't happen. We does not let it happen. Okay, we do not let it happen. So this is the method of changing the genotype of plant by either inactivation of the gene or sometimes knocking out the whole entire gene. Now, antisense RNA creating pest-resistant plant. Now, there are several nematodes. Now, see what happens. We have discussed how certain insects are infecting the plants, right? When we talked about the BT, cotton and everything. Now, we are going to discuss how a worm infects the vegetation and how do people with the usage of biotechnology cannot, you can say, rely on the presence of insecticides, pesticides and all of that. Now, there are several nematode parasites that are roundworm. Nematodes are roundworms that infect plants, animals, human beings, right? They infect all of us. Now, when they attack the plant, they feed on the root cells, which causes the root to grow into large knots or galls. These, like, they create tumors in the roots. Okay, tumors, big mass of cells. Now, this damages the crop. It does not let the root absorb enough water, enough minerals. Yes, Mariam, you wanted to say
How
something?
do they do that?
How do they do that? They start feeding there. They start dividing there. They start releasing certain enzymes to damage the root cells. So this damages the crop and reduces the yield. So that is why we call them as the root knot nematode. Now, a particular nematode called as the Melodigyne incognita. Okay, just 1 second. Yes. So there is a particular nematode called as Melodigyne incognita. Okay. They infect the root of which plant? So tobacco plant, not any other plant. So they are host specific and they cause a great reduction in the yield. So they are a great pest, a great threat to the vegetation of the tobacco plant. Now, the most effective and the sustainable management tactics for preventing such a damage is to engineer a resistant plant, just like how we made a plant resistant to the bacillus, right? Same way, we are now going to make a plant that is resistant to Melodigyne incognita. And this, you can say, process that we are opting for is RNA interference or RNAi. Now, this is a naturally occurring mechanism. Okay, this happens in many organisms. This is a natural mechanism. And this leads to the silencing of gene, meaning no translation will take place, meaning no proteins will be formed, right? No proteins. This is what is the meaning of gene expression. Okay. Now, in nature, this mechanism is used for the regulation of specific genes, and it is applied as a defense against viruses also, because this is occurring naturally. Now, it takes place in all the eukaryotic organisms as a method of cellular defense. So, whenever the cell wants to defend itself against the, you can say, external pathogen, okay, so the cell performs this particular defense mechanism. Now, in this particular case, wherein we talk about the silencing of mRNA. Now, you people know, tell me one thing, is mRNA double-stranded or single-stranded? Students, this is a very basic question. It's not even a thing that you people have to think about. It's not
Single
a tough.
stranded.
Yes, they are single. You people remember we used to draw mRNA like this?
Yes, ma'am.
So you people remember everything when I tell you. Huh? So the mRNA is single-stranded. That's true. The only double-stranded thing is the DNA, isn't it? So now the silencing of the mRNA, it takes place due to the formation of double-stranded mRNA. Meaning one will be the sense strand, one will be the antisense strand. Because of this very reason, the gene will not get expressed. The silencing of gene will take place. So due to the formation of double-stranded RNA molecule, formed by binding of complementary RNA, meaning, for example, normally every organism has their own mRNA which is synthesized by transcription process, isn't it? So then what happens? One mRNA is like one strand is there, and now during this defense mechanism, another strand opposite to that already existing strand is made. That is what we call as RNA, or you can say antisense RNA. So complementary RNA, or we can call it as antisense RNA. Okay, meaning, for example, I will show you in two colors. So for example, this is the normal mRNA. Okay. Normal cell mRNA, normal ones that the organism has. Now, when this particular antisense mRNA is made, so this will be made like this. Okay, and this antisense mRNA will cause the silencing of the mRNA. Why? Because the mRNA does not express itself in a double-stranded form. It is single-stranded, and that is only how it will be, you can say, expressing itself. Clear? This much is clear? Does anyone have any doubts with the concept of how it happens?
No, ma'am. Yes, clear, ma'am.
Okay. So it prevents the translation. Again, I told you that translation is the expression of gene because proteins are formed. So it prevents the translation of the original mRNA. Clear, students? This much is clear? All right. Now what happens in this particular, now how do we do this, right? How do we make a plant that can do all of this to prevent the, like, or not to prevent, to cause the gene, or you can say silencing of gene in melanogyna. Meaning in the parasite, when it eats the plant or when it tries to eat the plant, the mRNA silencing takes place in its body. Just like how when the bacillus BT cotton was there, the insect that came along to eat it, right? This thing, cotton corn borer, cotton bollworm, all of those. So they got perforation in their intestine, isn't it? They got the toxin in them. So same thing here. Now in this particular case, tell me one thing. What is the vector we use for plants? Like when we want to deliver the foreign gene into the plant cell, which vector do we use? You only have to tell me. I'll not fill that line. I can give you one alphabet. I'm not cruel. Now tell. Students, now this is not even so far of a chapter. It's the chapter we did before this chapter. No one remembers. Now you people check in your notes and tell me. No one found it. मिला भी नहीं, notes में मिला भी नहीं. What about the other students? None of you remember? None of you remember the vector that is used to deliver a foreign gene into the plant cell? You people remember retrovirus was for animal? Which one was for plants? That's my only question for you.
Agrobacterium
Yep, agrobacterium.
tume.
What is so special about agrobacterium?
It is the pathogen of several dicot plants, which is able to deliver a piece of DNA to host cell.
But what is the property of agrobacterium? Have I taught you that? Like, why is it used as a vector? Does it have some unique property?
Cloning. Cloning.
Cloning, but it also forms tumor. You remember TI plasmid?
Yeah.
Tumor inducing.
Tumor inducing.
They induce tumor. That is why they are used. Now everyone remembers. See, you people don't revise, you people don't study. Do you open your notebooks after the chapter is completed? Anyone who goes back and sees, okay, what we have done previously? No one. Are you those are you a part of those people that while watching a new episode you don't watch the recap? You like skip. Huh? That's what you do. It's very bad though. I'm smiling, but it's bad. All of you should revise the notes. And this chapter is like interconnected biotechnological processes, and now we are applying it. So you people should remember the terminologies the least. Now we're going to see the biotechnological applications in the field of medicine. Okay, this was all there is to know. The plant vector that we are using to deliver the gene of interest into the plant is the agrobacterium vector. So the host specific, or you can say the nematode specific genes are sent into the plant, and this causes RNA interference, just like how I told you. Now, when we talk about the biotechnological application in medicine, so in this case, what happens is that there are it has such a vast positive effect in terms of medicine. Okay, it has been great in the field of healthcare. So we are going to see how does that actually happen. Now, the biotechnological application, when we talk about healthcare, okay, healthcare has been better. Okay, it has been, you can say, improved because of the development in the biotechnology. Okay, so why? Because the first thing is that with the help of biotechnology, you people have learned about fermenters, bioreactors. You remember bioreactors? No, none of you. See that also. See in your notes. What is a bioreactor or fermenter? Those were the big vessels that had 100 to 1,000 liter volumes, right? In which large scale production of anything that we wanted to do was done, isn't it? So those were the fermenters, or you can call them as the bioreactors. So with the application of biotechnology, large scale production of medicine could be done, isn't it? On a huge, huge scale. Apart from that, since it directly affects the particular pathogen or the gene, so this has a great thing of having very less side effects. It's safe to use also, right? Because downstreaming processes are also done. So the recombinant DNA technology have made great impact in healthcare. Great impact in healthcare. Now talking about what are the advantages that it has in the field of this particular medicine or healthcare. The first advantage is that it enables mass production. Okay, meaning production in a huge quantity. That is, you can say, required by the public, right? Then second thing is that safe and effective drugs. Safe and effective drugs are made. And lastly, the third is that they do not have any kind of side effect. The reason why? Because they are targeted, isn't it? They are targeted to a particular gene, to a particular problem. So, no side effect. Clear? What is the spelling of side effects? SIDE, effects. This is how you write? That also you don't know. Tell me if it's right or not.
I think it's right. I just
Okay,
don't
just 1 second,
read
students.
that.
All right. Yeah. Now see, when we talk about how many drugs or how many medicines have been made, so all in all, there has been 30 recombinant therapeutics made. Out of those 30, 12 of them are being marketed in India. So a lot of it has been very successful, isn't it? So total 30 recombinant DNA technology therapeutics. This is like a fancy word for medicine, isn't it? Now, medicine could be injections or anything of that sort. So, are approved globally. Okay, it is approved globally, meaning it is safe for human use. And 12 of them or 12 of these are marketed in India, meaning presently being marketed. Just 1 second, students. Yes. So out of the 30 recombinant DNA therapeutics that have been approved, 12 of them are presently being marketed in India. Okay, meaning in India it has been used. Students, apart from these two, where are the rest of you? None of you have switched on your videos? Apart from Yahya and Maria, where are the rest of you? And I'm not even getting responses from you also. Am I only teaching 2 students in the class? Students, am I audible to you? This is not nice. Then why do you people come to attend the class only when I have to teach just these two? You people show no interest in studying. I don't know why. You are in class 12th. बड़े हो गए हो भाई अब. Behave like a 12th. And if you can't switch on your video, at least you can respond by using your mic. That is there. No one can see you when you switch on your video. Okay, tell me what is the last thing you people have written? This root knot?
Yes, ma'am.
Okay, now you people can note it down.
Done.
Done.
Everyone's done?
Done.
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 Right, all of you are done?
Yes, ma'am. Done, ma'am.
Done, students?
Yes, ma'am.
Done, ma'am.
Better, ma'am.
All of you are done?
Yes. Done, ma'am.
All right, everyone's done?
Yes, ma'am.
Okay. Now see, I'll give you some of the examples of the drugs that are approved in India. This is not something you people have to learn as such, but it's okay if you know, like remember it. And it's fine if you don't also. So the first drugs, the first one is human insulin. Human insulin. After human insulin, then comes along streptokinase. This is also synthesized with the help of RDT. Then comes erythropoietin. These are the names. Okay, you don't have to learn. You just should know. You should have this much, extra knowledge also. So this won't come in the exam. I will not ask it in the exam. And hepatitis B vaccine. That is also. You people remember hepatitis B?
Yes, ma'am.
Yes, ma'am.
It's an
Which
RNA
kind of
virus.
disease is it?
Incurable.
Incurable? What kind?
Uh,
What is the nature?
RNA virus.
No, no, no.
STD.
Mhm. Like
STD.
what? STD. Very good. Exactly. Sexually transmitted disease. Okay. So human growth hormone, that is also, you can say, engineered with biotechnology, and there are many. This is just like out of the 12, I have named five of them. All right. Now, we are going to talk about the recombinant insulin or the genetically engineered insulin. Very, very important. Recombinant insulin or genetically engineered insulin. These are like the interchangeable names that you can use for it. Both of them are correct. Now, when we talk about the genetically engineered insulin, now do you people remember which organ synthesizes the insulin?
Pancreas.
Pancreas.
Pancreas. Very good. Do you remember the specific cells?
Alpha and beta, right?
Exactly. They have alpha and beta. They have two types of cells. One is alpha and one is beta. Now the alpha synthesizes what and beta synthesizes what?
Glucose
Alpha synthesizes glucagon
and
and
beta insulin.
Insulin.
insulin. Correct. Now, do you people know what is the function of glucagon and insulin?
That I forgot.
See, when you talk about insulin, so insulin is the hormone also that is released in our body. Apart from that, insulin is also responsible for, you can say, breakdown of sugar or breakdown of blood sugar. Whereas insulin decreases the amount of sugar in our body and glucagon increases it. So this is responsible for controlling the level of glucose in our body. So these hormones, and you people do know that these are hormones, isn't it?
Yes,
So
ma'am.
these
Yes.
hormones, they are responsible for regulating, or you can say controlling or regulating the level of glucose in our body. The level of glucose in our, or you can say in our blood, to be very specific, in our blood. Now we know that what happens, students, when there is an insulin deficiency?
Diabetes.
Diabetes. Do you know the particular name of diabetes?
Like diabetic 1 and diabetes 2.
The diabetes mellitus. Okay, that is what the name is. Okay, clear, students?
Yes, ma'am.
So diabetes mellitus is the disease that is caused when there is insufficient release of insulin in our body. So because of this, the person who has the deficiency, they have to take in the insulin injections. Okay, they take in insulin injections so that this hormone is present in them. So that this hormone is responsible for the breakdown of glucose in their body. So we can write that a deficiency of insulin it causes a disease called as diabetes mellitus. So to treat it, insulin injections. Okay, insulin injections are taken by the patient, right? So insulin injections are taken. Now, what used to happen is that insulin, which was used for diabetes, very earlier, when it was synthesized for selling, so it was actually extracted from the pancreas of the slaughtered cattle and pigs, meaning the cattle, meaning the cows, the buffaloes, all of these that had been slaughtered, killed, or the pigs that have been slaughtered or killed. So from their pancreas, the insulin was extracted. Okay. Now, even and then it was sent to the human being's body. But since it was, like, you can say, extracted from the body of organisms like cows, buffaloes, pigs, so like it did work on the humans. It's not that it did not work, but then some people got allergic from it and some people did not took it because of the religious sentiments. So then insulin, and one more thing is that why not insulin is taken in any other form other than injection, students? Any idea about that? Why don't the people that have diabetes, they take insulin pills or anything of that sort? Why do they use the
Because it should come
injection?
because it should come directly from bloodstream, contact with bloodstream.
Hmm?
Because it should have a direct contact with the bloodstream.
Direct contact in the bloodstream. So if a person ingests the insulin, meaning takes in the insulin, so then also can be supplied to the bloodstream, isn't it? Like just like how our food is also supplied. What do you think? Why? What is the reason that the insulin can't be taken orally? It's very simple reason. Hmm? Anyone? No one. What is present in our stomach?
HCl.
Hydrochloric acid, isn't it? And if you make a reaction between hydrochloric acid and insulin, which is a hormone is a protein, so it will get degraded, isn't it? So that is why it can't be taken through the oral ingestion. So that is why injections were, you can say, created for insulin dependent diabetes. Okay. So it was extracted from the slaughtered cattle and slaughtered protein, I've written, slaughtered pigs. So what used to happen? This created a big problem as it became allergic to some patients, or as it caused allergy this much is clear, students. Now you people understand why the need of genetic engineered insulin is required.
Yes, ma'am.
Okay, now you people can note it down. Done, students?
Yes, ma'am.
Done, ma'am.
Done, ma'am.
All right, everyone's done.
Yes, ma'am.
If you are done, students, any doubts up until now?
No,
Anything
ma'am.
that you did not understand till now? Are you finding this
No, ma'am.
chapter nice now?
Yes, ma'am.
Yes.
Okay, now you are able to bear it, isn't it?
Yes, ma'am.
Yeah.
All right, very nice to hear that. So, do you have any doubts in all of the things that we have studied today?
No, ma'am.
No, ma'am.
Okay, the RNA interference is clear?
Yes, ma'am.
Yes.
All right, why the insulin needs to be genetically engineered, that is clear?
Yes, ma'am.
Yes, ma'am.
Okay, all right. Okay, students, then if everything is clear, then we'll keep it till here only for today's class. And in the next